Hello class, this is chapter 3, part 3. We're going to be talking about lipids. So lipids structurally do not look very similar. You know, the carbohydrates, it's basically one, two, or many of these six-sided rings linked together. Or possibly five-sided rings. But with lipids, each category looks a little different. And you have four categories. Those are fats and oils, phospholipids, steroids, and waxes. And you'll say, Dr. Hilton, that's 5 categories, but fats and oils are the same thing. They are structurally the same and functionally the same. So each of those groups looks a little different. Phospholipids and fats and oils are related. There's only a small difference. But the other two are very different. The thing that unites them, the thing that classifies something as a lipid, they are large and nonpolar, which means they're not soluble in water. So this is the thing that sort of defines a lipid. You know, with carbs, it was their CH2O ratio. Functions, I'll actually go over those as we go through each type of lipid, so don't worry about that just yet. Okay, so you can group fats and oils together and just call them triglycerides. And they are long-term energy storage. Triglycerides actually have more energy than glucose does, but your body again uses glucose first, so the fats and oils don't get burned as often. And your body tends to hoard triglycerides, so only burn them if you force it to. And the way to force it to burn fats is by having long periods of physical activity. If you starve yourself, your body starts eating proteins instead of glucose, which doesn't solve your problem. It will also eat fats, but again, you're destroying most of your protein going that route. So their function, though, is still long-term energy storage. They have the highest energy density, so per mass they have more energy than glucose. You can also call them fats and oils. Besides the energy storage, they have some protective abilities, both by insulating the body and by cushioning certain parts of the body. Your kidneys in particular are cushioned by fat tissue. The way we build them is we have 1 glycerol and 3 fatty acids. And even though they're not a true polymer, they're still linked together by dehydration synthesis. So it's 1 glycerol and 3 fatty acids. The tri here means 3, and glyceride is talking about glycerol that's linked to something. So the name tells you how it's built. The fatty acids in triglycerides can either be unsaturated or saturated. Saturated means there's no double bonds. Their chains may zigzag, but they're relatively straight. Because they're straight, they stack well. Because they stack well, they tend to be solid at room temperature. Some examples are butter and lard, and they're commonly from animal sources. Unsaturated fatty acids have at least one double bond. They will also zigzag, but due to the double bond, they'll have a bend. And because of that bend, they don't stack well. Because they don't stack well, they're liquid at room temperature. And they're commonly found in plant oils, but they're also in cold water fish. In general, you want to try to replace saturated fats with unsaturated fats, and you want to avoid trans fats like the plague, and I'll show you the difference. So here is a saturated triglyceride. Here's a saturated fatty acid. You'll notice in this long carbon chain there's no double bonds, and therefore it's straight. This is an unsaturated triglyceride because it has several different unsaturated fatty acids. It has a double bond here and a double bond here. Notice the hydrogens on either side of this double bond are on the same side. That is a cis. Cis literally means same. And so that is a cis double bond, and those are natural and they don't upset your body. However, on this double bond, the hydrogens are on opposite sides. That is artificial. Instead of a bend in your fatty acid, it is straight. And trans means across, so these hydrogens are across from one another. And those trans fats really inflame your body. They really upset your body. So that's why we want to avoid trans fats. Trans fats used to be in margarine, but since we learned that trans fats are horrible, they've pretty much gotten rid of it in margarine. You can still find them in vegetable shortening. I think it's Crisco that's vegetable shortening. And then in long-term baked goods, so those Twinkies and Ho-Hos and cupcakes that are supposed to last, until 2057, those also have several trans fats. So those are the foods you really want to avoid. But again, unsaturated fats, they're typically from plant sources. They're a little bit better for you. They have these crazy bends due to the double bonds. And that means they don't stack well and they're liquid at room temp. So I want you to think a little bit about coconut oil. Sometimes when you go to the store, the coconut oil is solid at room temp. Sometimes when you go to the store, the coconut oil is liquid at room temp. It's very sensitive to temperature changes. So does that mean it's a saturated fat or an unsaturated fat? It's from a plant, So it's probably unsaturated, but it is often solid at room temp. So which is it? Well, it probably... So glycerol, you can put any 3 fatty acids on here. So you could have a mixture of saturated and unsaturated fatty acids, and that's what's going on with coconut oil. So it's mostly saturated fats, but it also has some unsaturated fats, and that's why it's very sensitive to temperature changes. Sometimes it's solid, sometimes it's liquid. So here's glycerol. It is a carbohydrate by itself. And each of these OH groups are reactive, and we have 1, 2, 3 of them. So that means we have 3 groups that are reactive where we can hook on something functional like a fatty acid. So here's our glycerol, here are our fatty acids. Notice the fatty acids can differ in the length of the chain, how many carbons there are, as well as the double bonding. So where double bonds are present or not. And remember, when there's a normal cis double bond, you get a kink in the chain or a bend. We're going to link all 4 of these molecules together, these two, and these two, with dehydration synthesis, and we can break them with lipolysis. So again, this way is dehydration synthesis. We're making these 3 covalent bonds, and then we break them with hydrolysis. Alright, besides recognizing the reactions, you want to know that these are fatty acids. And again, you can tell by the long carbon-to-carbon linkage there, and there's the acid group. And you can see that even though these fatty acids are different, they still follow that same pattern. Fatty acid, or the acid group, and then the long carbon-to-carbon chain. They are actually similar. And then be able to recognize that this is a triglyceride. So 1 glycerol and 3 fatty acids all linked together. Alright, phospholipids are next. They are not for energy. They make membranes. Structurally, they're similar to triglycerides, but instead of 1 glycerol and 3 fatty acids, it's 1 glycerol, 2 fatty acids, and a phosphate group. So remember, glycerol has 3 slots where you can add stuff. So 2 of those are taken up by fatty acids, and the 3rd slot is taken up by the phosphate group. The fatty acid parts are nonpolar and not soluble in water. They don't mix with water. Hydrophobic is a term that just means not soluble in water. The hydro part there means water. And the phobic part means fear. So water fearing. The phosphate group is usually not just polar, but also ionic. So it's charged. Therefore, it's hydrophilic. Hydro again means water. Philic comes from philia, which means love. So polar stuff is hydrophilic, which means it's water soluble. It will mix with water and stuff like that. So that's the construction, that's the structure. The function is forming membranes within cells and the exterior membrane of the cell. And you form a phospholipid bilayer. And the polar part, the hydrophilic part, is oriented outside. And the nonpolar part is in the inside. And so here's a cartoon of a phospholipid. But if you look within the cartoon, you can see glycerol. You can see these fatty acids, a bunch of carbon-to-carbon, a long carbon-to-carbon chain, and there's a little bit, the remainder of that acid group. So you got two of those, and then there's the P, and it's actually a PO4, which, remember, that's a phosphate group. So those are all the structures that you expect to see. When you put a bunch of these phospholipids into solution, again, they form a phospholipid bilayer. Here's one layer, here's the other. Notice the heads are on the edge or the outside of this layer, and your tails are on the inside of this layer. Outside the cell is water-based. If it's in a body, it's going to be tissue fluid. Inside the cell is also water-based. The water-based part we call cytosol or cytoplasm, but both of those terms mean roughly the same thing, and they're both water-based. So you have water on the outside, water on the inside, and that's why the hydrophilic parts are located on the outside, where they can mix with water, and the hydrophobic parts are on the inside. These hydrophobic parts are important, however, because if water or anything polar or anything ionic tries to cross this lipid layer, it cannot cross. It is deflected. Alright, steroids are the next group. I really want you to focus on. I just want you to know a couple things about waxes. But again, know their structure and their function. So steroids they also have rings, which might be a little confusing with the carbs, but their rings are fused, and there's always 4. So it's not 1 or 2 like the carbs, it's not many like the carbs, it's always exactly 4. And then those 4 fused rings have modifications added to them. They're all based on cholesterol. And cholesterol can be structural, but many of the steroids are hormones. And we can also use cholesterol to make bile salts, which are found in bile, and they're based on steroids as well. But the main function of steroids are they're hormones, and hormones are chemical messengers. So they're about the body communicating with itself within the blood. So here's our basic steroid, cholesterol. And here's what I mean by fused. So this wall is part of this ring. It's also part of this ring. And this wall is part of these two rings. And this wall is part of these two rings. So that's what I mean by fused. And then you can have, if you look at the accoutrement, these accessories, they can change, but the 4 fused rings does not change. So you can see there's very little difference between estrogen and testosterone, even though they create very different effects in our body. Anyway, so for steroids, just focus on their structure, the 4 fused rings, and their function. Most of them function as hormones, which are chemical messengers. Waxes, just know where they're found, and they're all structural. They mostly function for protection. You can find it in the outer layer of plants, and then beeswax and earwax. They're really long fatty acids with an alcohol group. But that's pretty much it. Waxes function for protection. And you can find them in earwax or plant cuticles, and they're made of really long chain fatty acids. So that's it for them. And that's the end of our lecture on lipids. So let me know if you have any questions.